And then serotonin is more responsible for things like this, things like the dendrites and spines. Remember, we extracted a specific spine. That spine will mean the same thing every time. Every time a spine receives a connection, it's the same meaning every time. So serotonin's role with the specific cell is more of this green area, more of these connections, more of the axons and things. Remember that make up the neurophil that provides communication. So serotonin and thyroid are the two biggest factors here for cell development. And it's a little bit unknown. Mainly, I think, because of this little epoch here before delivery isn't studied enough. It's just not well understood. But we can see like the studies from UC San Diego about the size of the brain organelle. UC San Diego early last year in 2024 now, the size of the brain organelle will predict severity of socialness in the autistic child. And they found this on 2-month-olds, roughly 2 months old. It's UC San Diego. They can have like this ability to track that specific child to ages 3, 4, whatever. And does the specific child with the altered brain organelle size have severities with socialness, and are they so-called fit into this autistic phenotype? It's similar to the study that I talk about frequently about the salience network. Remember, that's what these were. You have the ACC, anterior cingulate cortex, which is deep in the medial prefrontal cortex, and then the insula, which is lateral. These those are the two critical hubs for the salience network. These two regions here will bias our attention kind of more powerfully than anything. That's why they're the hubs of the salience network. And then you have other regions, mostly subcortical, things like VTA, the raphe, hippocampus, amygdala, so forth, things like that. So that study from UCLA with the salience network was found on 6 weeks old. The study from UC San Diego was found on 2 months old. San Diego does a lot of good research here, but nothing is really done about it. Ezekiel Ben Ari does a lot of good stuff here. He has an episode. He was one of my first guests. He's the one that found that, well, he's kind of famous for inhibition during this time frame is excitatory. Remember, we talk a lot about excitation, inhibition. That might be the central theme of the autistic phenotype. And during this period that allows for massive growth here, we don't have inhibition so much. It's all excitation up until like perinatal. So this role here with the thyroid and the mother is a huge risk factor for developing autism. It should be well known. It should be a checklist. The conflict is, do you take the hypothyroidism medication? If you want to mitigate the risk factors for autism, yes, probably. But our environments are horrible, absolutely horrible for synthesizing proteins like this. Why? Because of things like this, exposure to things like this. This isolated blue light will destroy and not allow these to kind of synthesize properly. And another thing about this is the myelination. Serotonin and thyroid has huge implications for myelination. So we just want to talk about that one specific cell, but you can extract any cell and it's going to have implications because serotonin is the master kind of architect. It's the Michelangelo of all of these connections, all of them, especially with sensation. And then T3 is the master kind of engineer, Michelangelo's right-hand man of developing this cell. Because this cell, if we're going to expand or create St. Peter's Basilica, this specific cell is a specific block.